All right, so we'll work on this exam paper. It's from the last exam session in March. There isn't still a mark scheme for the questions here. So that's why I made the answers based on previous exam years and previous mark schemes. First question talks about, what do you call it, gas exchange. They're asking us here to state the name of the gas exchange surface that's available or is found in humans. That's what we call the alveoli, right? So you exchange gases by diffusion in the alveoli of the lungs, okay? Now notice they said state two features. This means you're only supposed to write two. You need to choose the best two or the ones that you're more confident with. So they said state two features of gas exchange surface. This means what? This means you're supposed to talk about only the structures, right? Meaning that you can't talk about, what do you call it, ventilation. You can't talk about blood circulation. So it has to be, you have to refer to the structures found in the gas exchange, meaning the alveoli, and what would make those ideal for diffusion. So one would be what? One would be that the wall are short. So that like you have the wall of the alveoli, wall of the capillaries are both very thin, okay? Another thing is that the alveoli, there's a large number of alveoli, right? There is millions and millions of alveoli and millions of capillaries. So that provides large surface area. Third is the fact that the surface of the alveoli here is moist. Why moist surfaces are good? Because the moist surface here would allow the gases first to dissolve, and then that would allow the diffusion rate to be even faster, all right? So once again, you need only to write two. If you write three, they would just ignore the third one, right? So look, I'll say large surface area to do too many what? Do too many alveoli and do too many capillaries. Another one is what? Another one is that the walls are thin, walls of alveoli and capillaries. Third is that the surfaces are moist so that gases can diffuse, right? All right, in these questions that start with state, you don't need to give any explanation. You just need to make a list of items, just like the way we've done it here. All right. Next, they said on the diagram, label with an X the external intercostal muscles. So take a look here. Do you see those white things? Those are the ribs. So that's a rib, okay? And in between those ribs, what we have what we call the intercostal muscles. Now, obviously, the one to the outside is the external. So you just put an arrow. Either you write the name of the muscle, or you just write, they asked me to write an X. So I'm just going to put an X here, okay? All right. Now, next, they said, state the name of the tissue forms C-shaped structure in the wall of the trachea. So do you see those rings you have here? Those rings are not like complete circles. As the question says, they look like letter C. So we call those letter C-shaped rings found both in the trachea and found in the bronchi. Those are known as what? Yes, what do we call them again?
Cartilage.
Those are known as the cartilage rings. And those are essential. Do you know why this is important to have them here? They're important for two things. They are important to support, so that would allow that trachea to stay open. But being cartilage is not like hard like bone. Cartilage feels like the tip of your nose or your ear flaps. It would allow support and it would allow flexibility. So it would allow to keep it open. Plus, if you're swallowing and bending, that trachea won't break because it's quite flexible. So we write the name, cartilage, and then we state their function. So we provide support, keep them open, prevent them from collapsing, and it maintains flexibility. All right. Is there any question about the first page? Now, every time you see this sign, this means that the question is important. So I'm putting signs on the frequent questions. Like this one here, they ask you to describe the effect of on the thorax. Thorax means the chest. So what are the effects on the thorax, on your chest, for the contraction of the diaphragm? So let me remind you of this. That's the diaphragm, right? What happens every time the diaphragm contracts? It actually flattens, right? Like this. So that's what happens here. And that's important. You know why? Because as this flattens, you find that the chest gets bigger, right? The diaphragm contracts, it flattens, volume increases. Every time the volume increases, what happens to the pressure? Pressure decreases, and that drives the air inside our bodies. So that's the way we let the air enter our body. So first of all, flattens the diaphragm as it contracts, and then increasing the volume. Every time volume increases, what happens to the pressure? Pressure decreases, right? And as the pressure decreases, you know what's going to happen? That makes the pressure inside your chest lower than the pressure on the outside, okay? And that would drive the air inside our lungs, all right? Is there any question? No, right here, they're asking us, they actually gave us the percentages of different gases in the air. They told us that nitrogen is like 78%. And then they wanted us to identify the gas that is 21%. This gas is going to be what? This gas must be the oxygen, right? That is oxygen. And third gas, they said this percentage is just 0.04. However, it increases like 100 times with the air that we breathe out. So what gas is this one? This gas is going to be carbon dioxide, okay? Lastly, this said there is a gas that's actually variable depending on where you're breathing, right? And this gas gets more saturated as you breathe out. So what gas is this one? That is water vapor, right? That is water vapor. So here we go. So they're asking me now, after this, they're asking us to explain, like, why is it that the oxygen decreases, like the oxygen is way more in the air you breathe than the air you breathe out. And carbon dioxide is way more in the air you breathe out than the one you breathe in. So why is it that this number is greater than this number? And why is it 21 is greater than 16? The answer has to do with what? The answer has to do with gas exchange in your alveoli. Like basically, every time you take a breath in, what happens? Oxygen comes at high concentration in the air and it diffuses into your blood. CO2 goes in the opposite direction. So that diffusion is basically responsible for the increase in CO2 in the air you expire, decrease in oxygen in the air that you breathe in or you inspire, okay? Oxygen is a reactant in aerobic respiration. Carbon dioxide is a product, okay? So oxygen, what does it do? How does it get into your blood? Oxygen diffuses into the blood from high concentration. Where is the high concentration? The high concentration is here, the air in the alveoli. And then it is used for what? What is it going to be used for? It's going to be used for aerobic respiration, okay? This process also produces, so the reactant is oxygen, the product is carbon dioxide, which diffuses in the opposite way. Carbon dioxide is going to diffuse now from blood back to the air. And that air is the one that you will exhale, the one that you will breathe out, okay? All right, is there any question about this page? Question 2 is about human reproduction. So they gave us a picture of the egg cell and sperms during the time of fertilization. And they're asking us here to explain adaptations of the sperm and of the egg cell. And they wanted you to relate the adaptation to fertilization and development. So you need to write this, state the adaptation, and you see how is it related to fertilization and development of the embryo. So first of all, let's find common things that are between those two cells. First common feature is the fact that they both got the haploid nucleus. Why haploid is such an important thing to have in gametes? Because when the gametes fuse, you know what's going to happen? You would have diploid. So every generation, if this is 23, this is 23, then the baby will have 46. So every generation will have the same number of chromosomes. Other than this, what else do we have? We have in the sperm those mitochondria in the middle part. And those mitochondria continuously provide energy for the movement of the tail. Also, at the tip of the sperm, we got that acrosome, which is needed for what? That's the one that basically digests the jelly coating. You see that the egg cell comes with a jelly coat. We call it a jelly layer, jelly coat. And the acrosome is to digest it during the time of fertilizer. So like the sperm comes from here, right? Digest the jelly coat, and then it would deliver the nucleus, all right? The jelly coat also has a great function. You know what it does? The jelly coat is the one that basically hardens right after the time of fertilization. So if there were any sperms here, those sperms wouldn't be able to enter, okay? So again, acrosome does like this, digest the jelly coat, and then right at the time of fertilization, find that the jelly coat becomes thicker, harder, so no other sperm can enter, okay? So this is worth 6 marks. And as I told you, we need not to write the feature, we need to explain it. So you write, explain, write, explain. So let's start with the first one. It says both gametes contain those nuclei, they're haploid. And being haploid means what? Means that will ensure that the fertilized egg, you know what we call that fertilized egg? We call it the zygote. Zygote contains the diploid number, right? The full number of chromosomes, okay? So that's the first one. Then I'm going to talk about the middle part of the sperm. Do you know what's special about the middle part of the sperm somewhere here? That's the one where you have large number of mitochondria, okay? Why sperms are very rich with mitochondria? Because energy is there, right? Energy is released for the movement of the flagellum or the tail. And those flagella, like for what? Those flagella allow the sperm to swim till it gets to the egg cell, all right? Also, the tip of the sperm is special. Remember what is there on the tip of the sperm? Tip of the sperm got that, we call it acrosome. It contains what? It contains enzymes to digest the jelly coats around the egg cell. Next is jelly coat. What it does, that's the one that hardens, preventing other sperms from entering. Okay? Because if other sperms enter, then you wouldn't have that full or diploid number of chromosomes. Then fertilization will fail. Lastly, it's about the egg cell. The egg cell got these. You know what those are rich with? Those are like food storage, mainly fats and proteins. And those provide food, provide nutrients during like the first few days after fertilization. Like as soon as the egg gets fertilized, what it does, it divides by mitosis. Where does the energy come from here? There's no implantation yet. All of the energy comes from the cytoplasm, from the food stored inside the cytoplasm. So the egg cell contains food storage, right? And that food storage is needed for what? Is needed to provide energy. What requires that energy is the embryo, right? Like embryo is the last stage. It's needed for the cells to divide. And you know what we call this structure? Like the structure that looks made up of cells and it's empty in the middle. We call that the hollow ball of cells, right? So that's why we need energy there. Okay, how much should you write in these questions? You're not going to write all that much. You don't have time to write all that much, right? Okay, even if you know all about it, you can write 2 pages. Don't write that much because you need to catch up and finish the entire exam. So you're supposed to write 3 features and 3 explanations, 3 to 3, okay? Like you say, flagellum for swimming. You say food storage for energy, okay? All right, next, still in the same topic. Next, they're asking you here about process of artificial insemination. Do you remember this one? This one, like we have them together. We always ask this about, one is called AI, artificial insemination, and the second one is IVF. let me remind you of AI, artificial insemination. What do? They collect sperms from the male, the father, and what do next? They inject the mom with FSH. Do you know why they inject the mom with FSH? Because that FSH does what? That allows her to produce many eggs, many egg cells, right? We call that multiple ovulation. Right after this, now what is going to happen? We take those sperms we collected from the father and we inject them. Here there are two important questions. When and where? Like, when is it supposed to be injected? And where suppose those sperms will be injected? Now, let's start with when. When has to be around day 14, time of ovulation, okay? Because egg cells will be available only during that time. Where, what would be the best location to inject the sperms? The closer to the eggs, the better. So you have two options. Option number one is here, like the uterus. Second option is right in the oviduct. That would be ideal to increase the chances of fertilization. So again, we're trying to help these parents here because they fail to get fertilization naturally. So this AI, artificial insemination, would actually enhance or increase the chance of fertilization. Okay? So what am I supposed to say first here? Yes, please.
Sperm from the male.
And then inject the female with
FSH.
And then?
To cause ovulation.
Right? Cause like ovulation. What kind of ovulation if it's like she's producing many eggs at the same time? We say like multiple ovulation.
Yeah.
Then
insert the sperm into the uterus.
Or
the vagina.
Further is better. Oviduct.
Or the oviduct, yeah.
Yeah, because
around
that
would
put it, around the time of
ovulation.
Ovulation. Perfect. Very good. Thank you. Okay. Question now. Look what they said. Outline how the process of in vitro IVF, how this process differs from artificial insemination. So let me remind you of the differences. Ovulation, keep in mind that only in AI we have natural ovulation because IVF, we collect the egg cells right from the ovaries, okay? So IVF egg cells come from the ovary without natural ovulation. Note that we inject FSH in either method. So either method would require FSH because we want to produce more eggs. Where do we produce more embryos? There are more embryos produced during IVF, okay? Because we're collecting the egg cells, get as many egg cells as possible, so that will be more embryos. Since we're producing like many embryos, what are we going to do with those spare embryos? We could keep those spare embryos for later on, right? And then collection of sperms, this is common, right? In both cases, the sperms got to be collected. Implantation is also common, all right? No intercourse, again, that's what we're trying to help. So there will be no intercourse either methods. Fertilization in oviduct, which one you expect the fertilization to take place inside the woman's body? That would only be an AI, right? Because in IVF, the fertilization takes place outside the woman's body, okay? So again, I need to choose two differences here between the two methods. So first, I'll say egg cells are collected from the ovary in IVF. So there is no ovulation here. Fertilization and development in IVF, where does it occur? It occurs in the clinic. It doesn't occur in the uterus, okay? Now, embryos are produced in IVF. Remember, we produce way more embryos in IVF. And what do we do with those embryos? What do we do with those spare embryos? With those spare embryos, we actually freeze, okay? And that has a good advantage. You know why? Because we could possibly check and select for healthy embryos. We select only the healthy ones and we implant those ones, okay? Again, in such questions, you could write just two small answers. You say IVF fertilization outside the woman's body. IVF, there are more embryos, okay? Now, the next one is also important. They like to ask this question. It's about social implications related to fertility treatments. When they say social, this means what? This means you need to refer to things related to cost, related to family, related to population size, right? So take a look at these pictures. One important thing is the financial problems. Where would the financial problems arise here? First is the cost of treatment, because health insurance doesn't cover these things. These are not like covered by any health insurance. So the cost. Second is what? Second is those treatments cause multiple births. With multiple births, there's a high chance of what? It puts financial burden on the parents, right? Also, there is good things. What are the good things? First, and the most obvious thing is that it allows parents who wants to have children, it would help them to have children. There is some other issues here related to what? Related to the fact that we're making embryos, and some of those embryos could be like basically killed, right? They're not going to use all of these embryos. So some of them will be killed. Others would be what? Others could be used for research. If you have human embryos, spare human embryos, we could possibly extract some stem cells from them and use those stem cells for treatments. Okay. Some ethical issues related to choosing gender, because if they choose one gender on expense of other, because we have those embryos, some parents may choose to have always girls. They don't want to have any boy, right? So those would be like some ethical issues related to that as well. Okay. So what am I going to say first? I'm going to talk about two positive things. I'm going to say 3 positives and 3 negatives. So first one is like the most obvious things. We're helping parents to have children, okay? Which may help countries. Think of countries with declining populations like Japan, North Europe. These countries have, the population is decreasing. So that would help those countries to increase their population size. These treatments might be what? These treatments might be expensive, all right? So that's something to consider, and may lead to multiple births. So both are related to financial issues. There's also a problem with what? There is also a problem with those spare embryos, right? What are we going to do with those unused or spare embryos? They may be killed, or they may be used for medical research, okay? Very good. You may say also, I can't like include everything in this small space. I can say that the treatment may not be successful. If it's not successful, then that would put the parents, give them emotional problems, right? They build hopes, and then the whole thing fails. They waste money, and then the whole thing fails. Fatima?
Can I say that there are ethical issues against selecting embryos of specific gender?
Yeah, you can say this as well. I was trying to focus more on social because that would be the main thing. But again, ethical issues would also relate to the society. Can we say advantages when they ask implication? That's a good question. The word implication refers to both. That's the positive and the negative, okay? That's what it's meant by it. Implication doesn't mean bad things, okay? They would have said disadvantage or problems, okay? We're done with this first 2 questions. Is there any questions so far? Next question 3 is about plants. So they're asking you about transpiration. So they took leaves. Let me just summarize. They took leaves, they weighed the leaf at the beginning, and then they exposed them to different temperatures. Some were put in a cooler temperature, some put in hotter temperature, and then they reweighed after that period. They will reweigh the leaf and see what happened to the mass of the leaf. As you can see here, what do we see? We see like the mass of the leaf at different temperatures. As the hotter the temperature is, the lower the mass is, right? It makes sense, because if you take a t-shirt, wet t-shirt, put it in hot, it would dry up. And that's the same thing is happening here. So they said, explain those results. So take a look at the graph. Try to explain why is it that the mass of the leaf is decreasing as you put it into hot temperature. Now, there's an important thing related to the graph questions. If the question starts with explain and is worth many marks, it's highly recommended to start with a description. Like, although they did not ask me to describe the graph, they just asked me to explain it, but it's highly recommended, since it's a big question, to start by just one line to describe the graph. So what am I going to say here? I'm going to describe the general trend. what am I going to say? I'm going to say as the temperature increases, what's happened to the mass? The mass of the leaf is decreasing. Higher temperature increases what? What effect does the temperature have on the water you have in the leaf? Note that the water molecules or any other molecules, as the temperature increases, what happens? They gain kinetic energy, and that allows them to do what? That would allow those water molecules to change state. So we go from liquid state here to the gas state in the airspace. Note that the evaporation will have on the surface of those mesophyll cells. And eventually, once I have the water into the gas form, what's going to happen? The gas or this water vapor will just diffuse out of the leaf. Okay? So first thing, higher temperature increases what? Increases the kinetic energy of the water. This causes what? This causes the evaporation of water. And then what? Where will the evaporation take place? Usually evaporation of water in a leaf happens on the spongy mesophyll. So it takes place on the surface of the spongy mesophyll cells, right? Where will the water go? The water will move from here into the airspace, right? And then the water vapor would now diffuse out. So water vapor then diffuses out from where? It always diffuses out through the stomata, right? What we call this combination, always the combination of evaporation, then diffusion. This combination is what we call transpiration, okay? Lastly, what am I going to say? I have to stress on something. I have to stress that the loss of water vapor or the loss of water in general is the reason why the mass is decreasing, right? Sometimes we go into so much depth and we forget the most basic thing. The most basic thing here is that the reason why the mass is decreasing is because it's drying up. The leaf is drying up. Okay? Is there any question? They wanted you to state three other things, or sorry, one extra thing, actually, just one extra factor that would also influence rate of transpiration. Note that CO2 has nothing to do with transpiration, right? That's not photosynthesis, okay? So what would it be? It would be either, we can't say temperature anymore, so it's going to be either humidity, wind, or light, okay? Humidity is the only factor that would slow down the rate of transpiration. All right, so we say wind, humidity, and what do you call it? Light intensity. Why light has to do with transpiration? Because it would open those stomata. Those stomata, they would only open, the guard cells would only open the stomata if it's bright light. Good. State the name of what you see there in this picture. You don't have, this picture is like every other exam paper, you find it there. This picture is a picture of Xylem, right? And they're asking us like the usual thing. They're asking us describe how Xylem is adapted to do its job, right? Do its function. So again, what is special about Xylem? Xylem, as we always say, it's dead, right? Dead in a good way here, because dead means hollow, all right? And being hollow means what? Being hollow means it would allow easy flow of water, right? Where else? It has these, do you see those rings? They were provided, they were shown here in the exam paper. Why these rings are so important? Those rings are known as the rings of lignin. So those are made up of lignin. And lignin is such an important thing because lignin gives me two things. It gives me support, gives the xylem support, and it also gives waterproofing. So it makes the walls of xylem, makes them waterproof. All right. So first of all, I say xylem is composed of what? What is xylem? Xylem is composed of like vessels, right? And these vessels are very thick walls, right? Why walls are thick? Because xylem does not only one thing, it does two things. Xylem provides support and transport of water. There's also something else. There's also rings of lignin. And those rings of lignin provide extra strength and also provide waterproofing. All right. One more thing is that it's completely hollow. Hollow means there is nothing inside, right? So there's no cytoplasm, there's no single organelles, there's no nucleus, right? Why is it good to have nothing inside? Because that's how water can easily flow inside xylem. Okay. All right. I guess this should be easy because we've done it so many times, but even the next one here, that's something they ask every time, which is active transport. When do you write about active transport? You would always write about active transport whenever you see things related to ions, things related to the roots, things related to the intestine, epithelial cells. That's always have to remind you of that. It could be mostly about active transport. And this is a cliche. What you have in front of you is just something you need to stamp on the exam paper whenever you see it, just like always there. So how do I start? I say, I give a title. I say this by active transport. From where to where, Yusuf?
From lower concentration to higher concentration.
Very good. Low concentration in the soil. High concentration is going to be inside the root hair cells. Through what? Through this, right? You know what we call this? We call it a protein carrier. It has a specific name, right? Carrier protein or protein carrier. And that carrier protein basically moves materials from their low concentration. My example, the low concentration is on the top to higher concentration. And this requires energy from respiration through carrier protein found in the cell membrane using energy from respiration. Note with osmosis, we say what? We say partially permeable membrane, right? Here we say a carrier protein, okay? All right, so we're halfway through. Now we're going to go to question 4, 5, 6. 6 is very short. So let's start with what? Let's start with this one here. They're giving me what? They give me bacteria, and they want you to differentiate, right? Similarities and differences between bacterium and a, what do you call it? A plant cell. 1 second, I'm just reading someone. Yeah. So what am I going to say first? Similarities. They made it easy, really. They gave you the things. So I'll say similarity would be what? Cell wall. Similarity would be DNA. Similarity would be like cytoplasm. Those are all common, right? So I would say things like cell wall, say DNA, say cytoplasm, all are common, right? Ribosomes, even the cell membrane is also common, okay? Difference is going to be what? If you say cell wall in plants is made up of cellulose, that would count as a difference, right? Because although they both contain cell walls, but the cell wall is made up of cellulose in a plant cell. Plant cells contain what? Plant cells also contain organelles, like the real organelles, such as nucleus, mitochondria, chloroplast. Only bacteria contain what? Only bacteria contain like circular DNA, flagella, and a capsule. Bacterial cell walls are not made of, what do you call it, cellulose. They're made up of a different substance. You can call it moraine, you can call it peptidoglycan, but it's not like needed in our syllabus. Just say different cell wall. Okay, again, very important thing here. When you write the factor or you write the thing, you need to say what? You need to say, when you write in differences, you need to refer to what you're talking about. So if you say circular DNA, you have to say the bacteria have circular DNA, right? Make sure you refer to what you're talking about. Okay. Also, that next question here is important. Just one, I'm trying to answer someone. Do we need to compare? Yes, you need to compare. You need to write differences. You need to write like what is the difference between the two of them. Yeah. So about the cholera. Cholera and how would cholera lead to diarrhea? So event number one is what? Event number one is this, right? That's when the bacterium attaches to those epithelial cells of the intestine, right? Right after this, what would the bacterium do? The bacterium starts to release the toxin. So it attaches, releases the toxin. And what does that toxin do? The toxin starts to cause those epithelial cells to release salts, mainly chloride, right? So mainly chloride would be released here. And what does that chloride do? The salts and the chloride here in the intestine does something wrong, right? Which basically dries up the cells. It withdraws water out of the body cells. And that leads to what? That would lead to the dehydration and the diarrhea, right? So osmosis happens, and that would lead to dehydration, okay? So again, cholera releases what? Yes, what am I starting here with? Cholera releases toxin, causing a release of what? This would cause the release of chloride. And then what? This lowers what? This lowers the water potential in the intestine and causes the water to move like down its concentration, down its water potential gradient, right? And this results in what? This results in the dehydration, the diarrhea. Why would diarrhea happen? Because water has been lost from the body tissues into the feces. Okay. All right. Here's the question about antibiotics. Like they're doing an experiment, and this experiment is the one we usually do to test for antibiotics. So look at it. We usually grow bacteria. So you like make a mini farm of bacteria. How would you grow bacteria? Take a dish and put this substance we call agar. That agar substance does what? It's very nutritious for bacteria. So bacteria grow very quickly. Next thing, what are you going to do? Next thing, you bring discs, paper discs, and you dip those discs into antibiotics. So each disc here contains like a different antibiotic. And then what would happen? The antibiotic starts to do its work. It starts to kill bacteria. Sometimes some antibiotics are more effective than the other. How would you tell? Take that dish, put it at like constant temperature for like 1 day or so, or 6 hours, just like it has to be long enough. And then you see the results. What do I see here? I see that the antibiotic C has been like a failure, right? Because it didn't kill any bacteria. How do you know if it's killing or not killing? If you see like there's an area around the disc and that area looks white, this means that the antibiotic like D here has diffused out and killed those bacteria, right? We usually call this area around the antibiotic, we call it the clear zone, all right? So the clear zone is the area where bacteria got killed, right? So for me, if I am to choose an antibiotic here, I would completely forget about C because all bacteria are resistant to C. A is not really effective either, right? So either you choose B and the best one is going to be D, right? So again, the question is asking us more or less about the same thing. They are telling me first, like, why you should choose antibiotic E. Why E is like the most effective? What evidence do you have tells you that E is the best antibiotic? So what am I going to say first? I'll say E produced the largest. What did we call this area again? We call that area the clear zone, right? The area where bacteria got killed. So I'll say produced the largest clear zone, and which shows that what? Which shows that the antibiotic diffused from that disc and then killed or slowed down the growth of bacteria. Okay. Next question is, if you ask me something to study on the night of the exam, I would always tell you to study about natural selection. It's really, really very important. It's here they're asking about natural selection in bacteria, but anything to do with natural selection and evolution is very important. So look at the question. They said, explain how bacteria can become resistant to antibiotics. Like, where do bacteria suddenly become, or how do bacteria suddenly become resistant to antibiotic? This is more or less the way they ask you about, like, for instance, how do giraffes get a long neck? That's the same thing. How do you always start here? You would always start by, yes, Yusuf?
A mutation for antibiotic.
Very good. So you'd always start with a mutation. Do you see how I wrote mutation? I said random mutation. Usually they want you to understand that the antibiotic is not the reason why the mutation has happened, right? The mutation is random. Mutations happen 24/7. And it's just like by luck, sometimes those mutations are become useful, all right? So for instance here, you see what I have? That's the antibiotic. It's been applied on bacteria. By luck, one bacterium got like a mutation, and this bacterium is becoming resistant. What's going to happen? All of those bacteria that are non-resistant, they will die out. And basically, we did a favor for this bacterium. You know why? Because now it has all of the space, all of the nutrient for itself. So it starts to do what? That bacterium now starts to reproduce, increases in number, right? And as it increases, as it reproduces, it would pass on this allele for resistance to its offspring. So you basically created a whole population of bacteria that are resistant by applying the antibiotic. Okay? So once again, the antibiotic kills the non-resistant bacteria. Now the resistant bacteria have less what? The resistant bacteria have less competition. We made it easy for the resistant bacteria to increase in number by applying the antibiotic. What resources they would have? They would have like more space, more oxygen, more nutrients. And the resistant bacteria starts to reproduce rapidly. As they reproduce, what are they going to do? They're going to pass that resistance to the next generation. All right. So their offspring receive the allele for resistance. Why do we always say allele here? We don't say gene, because the gene has in two versions: an allele for non-resistance and an allele for resistance. So that's why I said the word allele. I didn't say gene. What we call this whole thing that we were talking about, we call this evolution. And evolution happens by what? Evolution happens by natural selection. Okay. All right. Any question? So it says here how to minimize the risk of antibiotic B, like this antibiotic, from becoming like useless. So many of the antibiotics that we used to take in the past have become useless. Why would antibiotic with time become useless, non-effective? Because bacteria grow mutations against those antibiotics. So how do we prevent this from happening? What's the ways that we can maintain or keep antibiotics effective? One of the best things to do is to develop new antibiotics. So pharmaceutical companies try always to develop new antibiotics. And for us, we could do something similar. We could change the antibiotics, like try not to use the same antibiotic every time. Also, do not take the antibiotic unless the person is really sick. So they shouldn't be taken unless they're really needed. One more is not to take antibiotic unless it's a bacterial infection. Antibiotics are completely not effective against viruses. Lastly, it's to complete the dose, right? Take the full dose. So the doctor told you to take these antibiotics for 10 days. You're going to have to take it for 10 days. Okay? So we need just one of these to be mentioned. So it could be like use antibiotics less often. Do not use antibiotics for the flu or viral infections. Make sure that patients complete the dose, develop new antibiotics, and use different antibiotics every time. You can use different combination of antibiotics as well, right? That's if someone is really sick, okay? Any question? Okay, we're near the end. We're left now with just two questions. This question is from the ecology chapter. It's mostly from the very last topic about conservation, but it starts with a small question. They wanted you to use the pictures to identify these as mammals. So what you see there in the picture that really tells you that this animal is actually a mammal. So take a close look. You realize what? You realize they got these ear flaps, right? Do you see another thing other than the ear flap that tells you also that these are mammals? Yeah, very good. Do you see the tail? Like there are hairs here. So that tells you also that there is these are mammals. Fur, ear flaps, these are always tell you that these are mammals, right? Again, it has to be something visible. So when they put you visible in bold, then you can't say that they have mammary glands or anything else. No, it has to be visible thing. Okay. Moving to the graph thing. They gave you a graph. It's like two graphs put in one. A graph about one animal here. That's the one on the picture on top, and another one for the one here on the right. I just put two colors just to make our life easy while we're talking. So I'm going to refer to this animal as pink, and this animal, I'm going to refer to it as blue. Okay. So first thing is calculate percentage increase in the number of that pink animal between 1990 and 2000. How do you do that percentage difference, percentage increase, whatever? Subtract, find the difference, and divide by the lower value. Why am I dividing by the lower value? Because the question says percentage increase. So you just need to go to the question, find that value at 1990. So what was the value at 1990? 1990, the value here is what? It's 75, right? Halfway between 50 and 100. How much was the increase? 2000, the number is what? The number here is like 2 squares above 250. This means what? This is 255, 260, right? So now what am I supposed to do? I need to find the difference. So I say, well, it's 260 minus 75. Divide by what? Divide by the lower value, which is 75, multiply by 100. Okay? So how much we're going to get here? The answer is 247%. Okay? This shows you what? This shows you that they've been like doing a good work here, because they took these animals and they're trying to conserve them. They're trying to increase the numbers. So for this animal here, they increased this number quite a lot, which is really good, right? Now, the next question, look what they said. They said, describe the data about the other animal, right? So the other animal here, which is the one shown in blue. So you really need to use what? You really need to use the y-axis on the right. They said, describe how the data, so describe the changes in the number of this animal. So what am I going to say here? I'm going to start, as always, with a general thing. So what do we see? We see that there is an increase. So there's like a steady increase. We reached the largest number here, and then we decreased rapidly. And finally, we decreased slowly. Okay? So again, you need to use those numbers on the right-hand side, because that one is the one that's referring to this animal. So again, number of an animal increases until when? It increased all the way until this year. Do you see that year here? That's like 2003. And then from there onwards, it was decreasing. So we had a decrease. It was like a rapid decrease. After that maximum number, what was the maximum number we got there? The largest number we got was like nearly what? It's like 34, isn't it? Yeah, no, actually 33 here. Okay, so I'm going to tell them the maximum number, which is like 32 or 33. Let me just make sure. That's 32 and a half, and it's 1 square. Yeah, it's actually 32. Sorry. And then the number of animals decreased rapidly till 2009, till here. And finally, then decreased slowly. We reached a very small number. We reached a very small number, like actually just 2 animals at the end. Okay? Any question? Someone is asking a random question. They're asking me, is magnification, if you don't have paper 6 in your school, should you study things about magnification? It comes in paper 2 and it comes in paper 4. So you should be, basically, there's nothing special about paper 6. Whatever was in paper 6 comes in paper 2 and paper 4. All right. Next is what? Next is it suggests the conservation methods that were used to increase the animal. Like how, if you have a rare animal, you want to increase its number, what are the ways you could do to increase its number? Usually with animals, we have one standard way to do it. Like if you have an endangered animal, let's say we have those white tigers are really rare. What do you do first? First thing you do is that you bring them into some safe place, like a zoo or a clinic, and you start to breed them there. Okay, so that's the first thing. Once you have enough number, let's say we have now enough number of those pandas, what are we going to do next? Once you have them in large number, you reintroduce them into their natural habitat. But you're not going to introduce them into a habitat, like a random habitat. It has to be controlled environment. Basically, you have to introduce them to somewhere safe, away from hunters, away from predators. So you control the place where they're going to be released. Okay? So again, what do we call it when you bring animals somewhere safe and you try to increase the number? We call this program, we call it captive breeding. Okay? What's the point of captive breeding? The whole point here is to increase the number, right? And then we release them into the wilds. Okay? We usually release them into somewhere safe, like a natural park or a reserve. And then this reserve has to be like watched. Basically, we minimize the number of competitive animals, like other animals that would compete with those pandas or whatever the animal is. And we try to make sure that there are no direct predators, because that will reduce their numbers. Also, what can we do? Also, we could raise awareness. That's like the best investment. You educate people about the importance of those endangered species, because if people realize how important these animals are, then they would actually protect them. Okay? We could even force this to happen. We can force the laws to happen. We can fine people or put people in jail for those who hunt those animals. Okay? Now, returning back to this graph, do you remember we reached just 2 animals here? We're coming to a very small population. What would be the consequence if you have a very small population of animals? What would be the long-term consequence to this animal? What would you expect on the long term to happen to an animal where only 2 of it is left? Like, for instance, here, if you have very small population, usually you end up having like bigger problem. You would start to have animals from the same family breeding with each other, right? That has a very bad effect, because that will cause the rise of genetic diseases, right? They always advise humans not to marry from the same family, because like cousins, etc., because that would increase the chance of genetic problems. Same thing would apply for animals. So we always try to do what? We always try to increase diversity by having animals from different countries. But in my case here, it wouldn't happen, because we're only left with 2 members of this species, right? You would have what we call lack of variation. There will be very little variation if there's very few members of this species. And always with less variation, there's less chance of survival. Like if some disease happens, then all the animals would be killed at the same time, because they're all directly related to one another. Okay? So what am I going to say here? I say with a small population, right? Size of the, what do you call it? Genetic variation decreases, right? Because members of the same family are likely to do what? They're likely to breed, since there are only few numbers left, right, in this small population. So you would really have small number of alleles, and the chance of genetic diseases will increase a lot in this case, all right? With less variation, always, whenever I write about variation, I write about adaptation. I say, well, now with less variation, there's less chance to adapt, and there's less chance to evolve, right? And this results in the long term, if this animal is becoming very less likely to adapt, it is more likely to become what, Yusuf? So on the long term, what would happen to the animal? It would become
extinct.
Yeah, it would become extinct, all right? Someone is asking, like, what would cause a genetic disease in this case? Think about it that way. Think about those 2 animals. They both should have more or less the same genotype, right? Because they're siblings. If they both have, let's say, B capital B small, and they bred together, because there's no other option, then it's 25% chance of having this offspring with B small, B small. And usually genetic diseases are recessive. They're more likely to appear if they're what? If they're like members of the family what got bred together. Okay? All right, so we're getting to the last question. The last question is quite easy. It's like the easiest one among all of what we've solved. They gave you a table about enzymes, and they just wanted you to write the function of each enzyme. So first one they said is pepsin. They wanted you to write, they gave you like a sample here. They said breaks down maltose. Here, what I'm gonna say like pepsin, it's to break down what? Breaks down proteins to amino acids. Where does pepsin work? Pepsin is made in the stomach wall, but it works right in the stomach, right? The other enzyme they asked me about was trypsin. Do you know where trypsin works? Trypsin and pepsin both are proteases. Trypsin works in the intestine, right? All right, intestine. The last question, or the last enzyme they asked me about, was the one that breaks down maltose. The enzyme's name is maltase, right? Maltase is made on the surface of the villi, epithelial cells, and it works again in the intestine, small intestine. Okay, So that's what I'm going to say here. I'm going to say trypsin, same as pepsin. One of them works in the stomach. The other works in the small intestine. Now, the enzyme that breaks down maltose, you know what we call it? We call it maltase, right? And it's only found one place in our body. It's only found in the small intestine, okay? You can refer specifically, for instance, for trypsin, you can say the duodenum, and you can say for maltase, you can say the ileum, all right? So it's not like you can refer specifically to the site of action, okay? Now, even that last part here is very important. It's about protein synthesis. So they gave me the whole process. They put like a whole page describing the process of protein synthesis. And they wanted us to label or to write the name of part A. You know what part A is? They said it's the one that codes for proteins. What we call this section of DNA that codes for protein. Yusuf? Yeah, very good. So you're supposed to write the name on the next page. I'm just labeling here, and then I'm going to write them again there. And they wanted us to label part C. Part C, you know what organelle is this one? That's the organelle that basically makes protein, right? You know what we call, that organelle is known as the ribosome, right? So going back there. Yes, we reached the ribosome, okay? They even referred to this. Do you see that part there? Just 1 second, some technical problems. All right, yeah. So they're gonna ask me later on about those things. You know what we call those building blocks assembled on the ribosome? That would be what? Those would be the amino acids, right? How do I know? Because they showed me a ribosome and they showed me like a chain. So those must be the amino acids. So again, part A, we called it a gene, and part C, we called it a ribosome. And then they said, describe the event that occurs in stage number 2. So stage 2 shows you what? Shows you something like a thread coming out from the nucleus, right? So what do we call that thing that exits the nucleus right after the first stage of protein synthesis? Yes, very good. We call that the messenger RNA. So again, we say messenger RNA. What is messenger RNA? Messenger RNA is a copy of the gene. So it's a copy of the gene. What does it do? Once it's made in the nucleus, once it's being copied in the nucleus, it exits the nucleus and it joins the ribosomes, right? And right there is going to be used like a template. It's going to be used like a recipe to make the protein. Note those colorful things you see here. Those are amino acids. The order of those amino acids is determined by what? What determines like which amino acid comes first and which amino acid comes second is the recipe, the code that we copied from the DNA. So once again, what am I going to say now? I'm going to tell them that I understand what's happening. Okay. I'll tell them that's the messenger RNA. Okay, so I'll tell them that leaves the nucleus to the ribosome. And where are ribosomes found? Ribosomes are found in the cytoplasm, okay? Before the last question, the state that what determines the order which part labeled B, the amino acids, what determines their order? The order of amino acids is determined by what? The order of amino acids, yes, what is it determined by?
The base pairs.
Very good. It's determined by the bases or the sequence of bases on messenger RNA, which was originally copied from where? Which was copied from DNA. Last thing here, still in the same like theme, it's about enzymes. They said, why is the shape of enzymes such very important thing? Like why enzymes have to be of specific shapes? Shape of the enzyme determines its active site, and that active site is where the substrate binds, right? So anything that changes the shape of the active site would not allow this enzyme to work, okay? So when the enzyme and substrate bind, what we call that? We call that the complex. And that complex is so important to cause what? To cause the, the formation of products, okay? So once again, we tell them here, it's the shape of the active site of the enzyme is complementary to the shape of what? Is complementary to the shape of the substrate, which is also important for what? which is important to make the complex, enzyme substrate complex. And without this complex, you wouldn't be able to carry out the reaction. The enzyme would fail to catalyze the reaction. Okay, right. So do you guys have questions? Any question about what we've done with this paper? Now, many of you don't do paper 6, so I'm going to be doing the paper 6 now. If you don't do paper 6 in your school, then you can leave now. So I'll shift now to paper 6 exam. It's taken from last year. It talks about what? It talks about what we call whisking tubes, mainly osmosis. Okay, so the question starts like this. They took some whisking tube. Again, I keep saying whisking tube. What it is, it's just like a bag, but that bag is special. That bag, the whisking tube bag, is special because it has tiny holes in it. So what are we going to do with it? We're going to fill it up with sugar, sucrose, and we're going to fill it up. We take 2 bags. One will fill up with sugar and one will fill up with water, all right? And then we're going to take those bags and put them into a test tube that contains only water, okay? So once again, take those whisking tubes, fill them up either with sugar. We're going to call that first whisking tube for that reason. What are we going to call it? Based on the question, we're going to call this one S, and this one we're going to call DW. DW for distilled water. We're going to put them into a larger test tube, and then we're going to wait for 15 minutes and see what's going to happen. Okay? All right, so that's the thing. Again, we take that tube, fill it up, tie it from both ends, and put them into a large test tube. Right, so here is the results. What happened? After 15 minutes, the bag that contains sugar, so the bag that had sugar inside, you see what happened? It got more volume inside. So like the volume inside this bag has increased, while the bag I only put water inside stayed the same way it is. Why is it so? What happened that made the bag here get heavier, increases volume of fluids inside, while the other bag stays the way it is? Reason here has to do with what? Has to do with osmosis. Like basically, look, this bag got sugar. The sugar inside the bag does what? The sugar inside the bag lowers the water potential, all right? And since the water potential inside the bag is lower, then what's going to happen? That drives the water by osmosis into the bag. So that made what? That made the volume inside the whisking tube increase. The other bag doesn't have any difference, right? There's water in, water out. So there will be no net movement of water, right? So movement of water will be equal on both sides. So if you wait 15 minutes, you will notice that there is no change in the content of the bag. Okay? Do you understand what happened? Now, they wanted you to make a table of results. They wanted you to write those results inside a table. So that they insisted on what? They said include data from both bags and make measurements about the volume before and the volume after. They wanted you to measure the length of the bag before and after. So look what I'm going to do. I'm going to make a table of 3 columns. The column of the left would be for the independent variable. What am I changing here? Do you notice what I was changing? I was changing the content of the bag. So I'm going to say here, tubing, or the content of the bag, or the concentration. And I write what? I write S, and I write the DW. Again, S in the question, they're referring to the sugar, sucrose, and DW refers to distilled water. Then we made measurements with your ruler, make a measurement from here all the way to the end, and make another measurement here, all right? So the first measurement here for 0 minutes, for here and here, it's nearly 10 millimeters, all right? Same would apply for the other bag. After 15 minutes, okay, they said that 15 minutes, the bag, this one got more size, right? So I'm gonna say like it's, that number is too big. I'm just gonna say like it's nearly 19, okay? I'm just assuming the numbers because I didn't do the measurement myself, okay? Then DW is not gonna change its size, okay? If someone has done the measurements, he could tell me here. They said, calculate the change in distance between those, the bag before and after. So how much is the difference in the size? you say basically 19 minus 10 is 9. So you write 9 here. And here what you're going to do, you say 10 minus 10 is a 0. Okay? Now, explain why it's important to take measurements from the same place. Like while we are measuring, why should we measure from the same starting point, the end point here, for both bags? Why do we decide to standardize the methods of measurements? This is very important in science. When I have two sets, I have to do standard way of measurements, because I want to do what? I want to make these results comparable. I want to have like a fair comparison between those two results, all right? Again, you don't say to make it reliable, you only say reliable. Look, the word reliable starts with RE, right? Reliable always refers to repeats, starts with RE as well. So that's what we have to keep in mind. I didn't do several measurements. I was just trying to compare between the two bags. So I'm going to say to have fair comparison between those two bags. State a conclusion. What would be the conclusion you can come up with here? Again, with paper 6, you really have to make your conclusions as basic as possible. Don't go into details. First, it's not worth it. It's just 1 mark. Second, it's core. That paper 6 is for core and extended. So you don't expect to come up with very detailed explanation here. So I'll write you 2 conclusions. One very basic and one like more advanced. So first one, I'll say what? First one, I'll just say the bag that got the sugar increased its content. So water entered the bag with the sugar. That would be worth 1 mark. Or if you want to write about osmosis, also possible. What do we say? You say that the bag that got the sugar here attracts the water in by osmosis, or lowers the water potential and water moves into the bag by osmosis. Any question? What was the independent variable in this investigation? Like what's the variable we were changing in this investigation? Remember what was the content of the bag? We made 1 bag with sugar, 1 bag with water. So we could say sugar, no sugar, or you can say the concentration of the content of the bag. That's the thing that we changed, right? So that's the factor that we actually changed, right? All right. And they said, explain why it's better to do several, like they said, 1 set of results collected during the investigation is not going to be good enough. Why should we do several sets of results? What's the point of doing several measurements? These questions are always fixed or standard in paper 6. If they ask you like why we did several measurements, what would you tell them? We tell them, well, it would help me to make my measurements reliable. It would help me to find errors, like anomalies means errors, right? It would help me to find the average, okay? If they ask you like why we found a difference, or why did we do a percentage difference, what would you tell them? We tell them like for fair comparison. If they told you why shouldn't be using like different droppers or different syringes, what would you tell them? You tell them because that would prevent mixing solutions and that would prevent contamination, okay? So learn those questions very well, because there will be asked. Sometimes they ask you this 2 of those questions in the same exam paper, like the one we have in here. So back to my question. The question is asking you like, why did we do several sets of results? What would you say here? We say to increase reliability and to find errors, find anomalies, okay? Or to find the average, right? Because if you do 1 trial and this single trial you've done was wrong, then the whole experiment would be wrong, right? To calculate the average, okay? Some other students, look what he's done. He was using 1 syringe only, and he got different results than us here. What would you tell this student who used like the same syringe for sucrose, and then he used that same syringe for water? What would we say here? We say that this syringe would have mixture of both solutions. It would have traces of both solutions. What we call this thing when you're mixing solution being messy, we say that this would result in contamination, right? So the sugar solution was mixed with water, And when you mix sugar and water, then that would cause a change in the concentration. All right. Any question? Also, food tests. Food tests are standard questions on the paper 6 exam. Here they're asking me, describe how would you test for the presence of reducing sugar. What do we usually use here? We use Benedict's, right? We add the Benedict solution, and then we put into hot water bath. Note that Benedict test doesn't work unless you put into hot water bath. How much is the temperature of the water bath usually? Is 80 degrees Celsius. Okay, so again, here's worth just 2 marks. So I'm going to say add Benedict, heat in a water bath. If it was worth 3 marks, you tell them like at 80, or you tell them like about the color change. You tell them like the color turns orange, red, green, or yellow. Okay. Next part, they said, well, they gave me whole instructions, and they said, state 2 variables that were kept constant. So what are the control variables in this investigation? What's the best way to find control variables? Always to find the control variables, find numbers, spot the digits, the numbers. Because if someone told you that we cut potatoes 2 centimeters long, that means they fixed the size of the potato. Or for instance, if they said, we did this experiment for 1 hour, this means what? This means they've controlled the time. Or if they said a potato, if they said like, we cut all of the cylinders from the same potato, this means what? This means also what? They've taken all of them from the single plant. So all of these are possible control variables. So I'll say size or the length, the soaking time 1 hour, and the fact that we took all cylinders from the same potato. All right. Again, that's the only calculation we do in biology, percentage difference, percentage change. They asked me about it here. They said, find percentage change for the last set of potatoes here. You don't need to find the change because it was given here. So I say the change is 0.43. How much is the initial value? The initial value is given here. So what am I going to do? I take that 0.43 and I divide by the initial value, 2.51, and I multiply by 100. So again, 0.43 divided by 2.51 times 100. Since the 0.43 is negative, then your final one is going to be negative as well. Okay, so negative 17.1. Okay, what does it mean if the value is negative? This means that the potato got shrunk, that got smaller in size. All right. Now, the reason I chose this exam paper particularly because the graph here is very unique. It's very rare graph because they asked you to draw curved line of best fit. Usually we're used to what? We're used to like point to point, a bar chart, line of best fit. But this time is like they asked us for a curved line. This means you're going to draw this with your hand this time. So first thing, I'll take that table of results and I show you how we're going to plot the results. So these are my table of results taken from the previous page. Okay. As usual, what do? We take the line on the left, that would be the X, and the one on the right, that would be the Y axis. Okay. So I'll take that X axis here. It's going to be, I'm going to copy here. So just going to copy this and put it down here. Copy it the way it is, just like the way I've done it. And write it here, 0.2, 0.4, 0.6, 0.8, till 1, okay? Next, I'm gonna take those values. Some of those values are positive, some of them are negative. So I'll take those positive ones. Do you see what's the largest number I have? The largest I have here is 7.5, and the largest negative value I have is 17.1. So look at the way I'm gonna divide it. I'm gonna divide this into 5, 10, positive, positive. That's the 0 value, right? I'm going to divide this into negative values. So getting negative 5, negative 10, negative 15, negative 20. So this basically ensures that all of the numbers I have here will be fit in my y-axis. And again, copy whatever the heading of the column is. So just copy it here, okay? Then plot. So we're going to plot the 0.2 here. So first the 0. 0 is 7.5. So this is going to go here. And 0.2 with 1.5. And 0.2 with 1.5. So somewhere here. Okay. You do all of the plotting. So I've already plotted it here. It'd be that way. And then with your pencil, you're going to join those dots together. So again, with one, don't move your hand from the paper. Join them all in one take like this. It doesn't have to join them all, but try your best to have them all in one take like this. And that would be what? That would be like a curve, just like what they asked you for. That would be a curve of best fit. Okay? Any question? All right, we're near the end. I'm just going to take a few more minutes, and then I'll let you go. Estimate the concentration of sugar, which there is no change in mass. So where's the mass graph? That's the mass. Where's the mass axis? That's the mass axis. No change in mass means what? How much is going to be no change in mass? That would be a 0. So where on your curve you had no change in mass? It's here, right? So somewhere right here. What are you going to do now? With your ruler, take this and see what exactly the concentration right there. Okay? So again, read how much is the concentration when there was no change in mass right here. How much is this? This is like nearly 0.24, yeah, 0.24, 0.26, somewhere in this concentration. How will the examiner mark this part? They only need to see the working. They only need to see that you've actually worked it out from your graph, and they will match it with whatever reading you have here. Okay? So not all of us will have the same number. It depends on your curve. But nonetheless, you will be rewarded this mark if they just found you've done the right work and you copied the right number. Okay? They said, explain why there was the mass is more useful than change of, why the percentage change in mass is more useful than change analyzing the results in table 1.1. Like in the first part of the exam paper, we were measuring length, right? Why change in mass would be much better way than like change of, why it's like percentage change in mass is better than the length we've done in the first part. This is another, again, there's another standard question. When you do something like this, you're basically making it standard, especially with potatoes. You cut them all the same length, but there's no way you will have them all of the same mass, all right? So that's why we need to find the percentage here, because you may start with some small differences in masses, like here, look at that table. The initial mass was never the same, all right? For that reason, it would be so unfair if I just find the differences. The differences would be very misleading unless I find the percentage. That's the only way I can have these numbers comparable, okay? This is something like if somebody done a test with 15 multiple choice questions and someone else done a test with 10 multiple choice questions. The first one scored like 10, the other one scored 10. Is this fair? It's not fair, right? Because that's 100%. And the other one did not score as high, right? So that's the same idea. When we find percentages, it would be a fair comparison. Okay? One more food test. This time the test is the starch test. How do you do the starch test? Easy, right? You take some drops of iodine, and if it's positive result, what would be the positive result? The positive result is like blue-black color. Okay? All right. Time for the drawing. Benedict is usually 80 degrees. With the iodine, you don't need to heat in the first place. Please, if you're not sure about your drawing, please send it to me, okay? So I'm gonna draw that tomato now. If you're not sure about if your drawing is right or wrong, just send it to me and I'll tell you if it's right or wrong. So here's that tomato they asked me to draw. So first thing to do, as always, is that you need to have a sharp pencil and draw an outline as big as the space you've been given, okay? So that's the outline, supposedly, all right? And occupy as much of the space that you've been given. And then what? Whatever is inside is still considered as an outline, right? So that is still an outline. I believe it's going to become very unsuccessful, but let's try. All right, so that's the inner part. It has a wall, right? You see that it has this wall. So you must include it, right? So I'm just going to put that wall here. Okay, so that's the skin, the outer skin of this tomato. And then what? And then I'm going to include those seeds. See, they're like, count those seeds, count how many seeds you have. Try to match the same number, nearly the same number of seeds you have on both sides. Okay, same thing here. I have seeds. And again, count them. And then what? You're almost done. There's a small detail here in this tomato. As you can see, they did not put it like direct. They've moved it on this side. So you really, one of the details you must include here is what? One of the details you must include is that curved part, the 3D part they've included here. So I'm going to include it in my drawing. Just show it here like this. All right. So that would be worth 1 mark in this. Again, the size and outline, proportions, I would lose 1 mark here for the proportions because it's not good, right? Because look at the wall. The wall is too thick. So I'll be losing this mark here. Details are what? Details would be the seeds and that curved part they have there on the end. So like ideally the drawing should look like this. Okay. All right. Lastly is before the last, they wanted you to test a tomato for vitamin C. How would you test for vitamin C here? You need the right indicator. So first, prepare an extract, prepare the juice of that tomato. You crush it. And what are you going to do next? Add filter it, make an extract. So you need to filter it. Take the juice and add DCP-IP to it. Note that we're not trying to find the concentration or the amount of vitamin C. We've just been asked for what? We've just been asked to check, does it have or it doesn't have any vitamin C? So what am I going to do? I'm just going to pour that DCP-IP drop by drop. And if it has vitamin C, what would you notice? You would notice that the color of the DCP-IP is changing. Okay? So you could do this either way. You could take the DCP-IP, put it at the bottom here, like put the juice here at the bottom, and drop the tomato juice from the burette on top. So that's one option. Or you could do the other way. So either way, you would notice that the color is changing. And again, you don't need to measure the volume. You don't need to find the concentration. You're just here to prove, does it have or it does not have? So crush and filter, put in a flask, add DCP-IP, and then see if the color is changing. If the color of the DCP-IP goes blue to colorless, then that would prove that it has vitamin C. Okay? Here they gave me 1 tomato seed, and they asked me to find the actual size of this seed. Note that this seed has been magnified 50 times. The length from end to end, as it's stated in the mark scheme, it's 106. Your value is going to be different, but let's assume it's 106. How do we find the actual? The actual is always the image, 106, divided by the magnification. Magnification is 50. So how much is it going to be? 106 divided by the magnification, 50. That would be what? That would be, we're just going to keep the number the way it is. So that would be like 2.1 millimeter. Okay. Why is it millimeters? Because we've done the first measurement in millimeters as well. Okay. Now we come like the largest question, which is the planning part. They didn't specify any significant figures here. So basically try to keep the same number of significant figures as the question is. In my case here, since I don't have numbers given here, I would just write whatever's there in my calculator. So here's the planning. They said plan investigation to find the optimum temperature for germination. Like what's the independent variable here? What are you going to change? The question says optimum temperature for seeds to germinate. So look at my plan. I'll get 1 tomato, take the seeds out of it, take a large number of seeds. And what am I going to do? I'm going to take those seeds and put them into like fixed mass of soil. I have soil, exactly the same type of soil. I'll put those seeds in the soil. And then what? Then I'm going to put every pack of seeds, I'm going to put it at different temperature because, again, what I'm changing here is the temperature. Temperature is an independent variable. You have to remember that seeds would not germinate unless what? Unless you put water. So also I'm going to control the volume of water. I'm going to put fixed volume of water every time. I'm going to wait. No, like seeds don't germinate in a matter of hours, right? Seeds take some long time to germinate. So it's going to be like a couple of days, a week, up to 2 weeks. So wait long time, state a realistic time for seeds to germinate. How would you know that seeds germinated? Seeds germinate when you start to see like the roots or the stem. So that's something also we should include. We should include the endpoint. We include like when is the time when the seeds, or what would be the thing we'll observe when seeds germinate. Okay, so look, I'm collecting a large number of seeds because that would help me to do repeats, so like 50 seeds. Where am I going to collect them from? I'm going to collect them all from one tomato, so tomato plants. And then I'm going to prepare what? I'm going to prepare water baths. Those water baths we always mention thermostatically controlled. I'm going to do it first at 20. And look, at the end of my planning, I'm going to say repeat at the other temperatures. Okay, so I'm going to take 10 seeds per water bath, and I'm going to put it into the sample of soil. I'm going to add water, fixed volume. And after what? After like a week, 2 weeks, I'm going to count the number of seeds that would germinate. That would help me to find a percentage number of seeds that have germinated. Okay. What will I observe? What will I be waiting for? How will I know that seeds have germinated? I would just see the shoot or the roots. In these questions, always, even if you've not been asked, mention a safety. Safety here is going to be what? Safety would be like wearing gloves because soil is dirty. Soil got pathogens in it. So you have to be careful. And write a conclusion. What would be the conclusion? Something like the optimum temperature is the one where more seeds germinate, or the higher percentage of seeds that germinate. Okay? Wear any safety thing. It doesn't have to be gloves. They're not really anything to protect yourself from the soil or irritation from the seeds, etc. Okay? All right. Sorry for keeping you late. I'm going to let you go if you don't have questions.